Hello, welcome to this lesson. In this lesson, we're going to look at enzymes. Enzymes are organic catalysts that are protein in nature and produced by living cells. They function to speed up or slow down chemical reactions in the cells and are thus said to be biological catalysts. Because a catalyst is a substance that affects the rate of reaction of a process. So enzymes are biological catalysts. Now enzymes are very important in that they speed up. So look at the importance of the enzymes. They speed up biochemical reactions in cells that would otherwise be too slow, or reactions that would not take place at all. Enzymes also control the reactions that are too fast, or reactions that produce large amount of heat energy to prevent them from burning and damaging the cells. So without enzymes, there are many reactions in the body that will be too slow to occur at the normal room temperature, or that will be moving too fast, producing so much heat that would end up damaging the cells. Now, there are two main categories of enzymes. These are, one, the intracellular enzymes. Now, intracellular enzymes are secreted and used within the cells that produce them. For example, most enzymes within the cells are intracellular, like respiratory enzymes, photosynthetic enzymes. These are produced within the cells and they function inside, where they control the rate of various reactions. The other category are the extracellular enzymes. Extracellular enzymes are produced within the cells like all the enzymes, but are secreted and are thus used outside the cells that produce them. For example, digestive enzymes. In many organisms, enzymes are produced within the cells and then they are secreted to the outside, where they break down food substances, and then the products of the food substances are absorbed into the cells. Now we look at how enzymes are named, because there are so many different types of enzymes, and it is important that they are named so that we do not confuse them. Now there are two systems of naming. The first one is known as the trivial naming system. Now, trivial naming system is no longer in active use because it was based on the persons that discovered and first described the enzymes. Some of the trivial names are still in use today, and they include enzymes such as pepsin, trypsin, and thiolin. These are trivial names that, though still in use, but those particular enzymes have been given modern names that uses a system of attaching a suffix ase either to the name of the substance upon which the enzyme works, or the type of reaction that the enzyme catalyzes. So the second system of naming is by use of the suffix ase. For example, enzymes that are named according to the substrate that they act on. We have, for example, amylase, amylase is named after starch, which in Latin is known as amylose. Sucrose is a substrate and the enzyme is known as sucrase. We also have the term protease to refer to all those enzymes that break down protein. For example, trypsin and pepsin are protease enzymes. Lipids is a substrate and the enzyme that breaks it down is known as the lipase. So apart from attaching the suffix "ase" to the name of the substrate, another way of naming the enzymes is by attaching the suffix "ase" to the type of reaction that the enzymes catalyze. So for example, if the enzyme catalyzes a hydrolysis reaction, then the enzyme is called hydrolase enzyme. And if it catalyzes an oxidation process, then that enzyme is known as oxidase. There are those enzymes that catalyze reactions that involves removal of hydrogen, for example, from a compound, a process known as dehydrogenation. Now such enzymes will be known as dehydrogenase, such that you find that there are many enzymes that are named according to the reactions they catalyze. Then there are those enzymes that are also named according to the substrate that they act upon. So the use of ASE is a modern way of naming the enzymes. Now next look at the properties of enzyme. Now enzymes are protein in nature. Being protein in nature therefore means that they are affected by temperature. They are sensitive to temperature, and also they are affected by pH. High temperatures would denature and cause coagulation of proteins. Many proteins are destroyed by high temperatures. In the same way, extreme pH would also destroy many proteins. Secondly, enzymes have a high turnover number. Enzymes have a high turnover number. Now, a turnover number refers to the number of substrates that an enzyme converts into products per unit time. So how many substrates are converted into products per unit time? It may be per second or per minute. That is what is known as the turnover. So turnover number refers to the number of substrates, molecules, that are converted to products per unit time by a particular enzyme. In general, enzymes have very high turnover. Now, an enzyme, like for example, there's an enzyme known as carbonic anhydrase found in the red blood cells. Carbonic anhydrase has a turnover number of 500,000 per second. That is, in 1 second, it converts 500,000 substrates into products. And in this case, it causes the reaction. It activates or catalyzes the reaction between carbon 4 oxide and water to produce carbonic acid. So in 1 second, it will convert 500,000 carbon 4 oxide and water particles into carbonic acid. Another enzyme, one that is very fast, in fact the fastest of all known enzymes, is known as catalase, found in many living cells. Catalase catalyzes the breakdown of hydrogen peroxide into water and oxygen. Catalase has a turnover of 6 million per second. Very, very high turnover indeed. So enzymes in general have a very high turnover. The third property is their substrate specificity. Enzymes are substrate specific. This means that a particular enzyme will only react with a particular substrate. This comes about because of the unique shape of the protein molecule that makes up the enzyme. Each enzyme has an active site. Each enzyme has an active site. This is the active site. This region here, that is the active site of the enzyme. Now, the shape of the active site is such that it's only a specific substrate with a specific shape that can fit and interact with this particular enzyme. For instance, we have two substrates here, substrate A and substrate B. It's only A whose shape at this point here matches the active site of the enzyme. So substrate A can react with the enzyme. If you look at substrate B, the shape is different, so it will not be able to fit onto the active site. So this enzyme is very specific to substrate A. It will only react with substrate A and not substrate B. And that's what you mean by each enzyme is substrate specific. So for example, the enzyme sucrase will only act on sucrose but not maltose or lactose because of the enzyme specificity, which is determined by the shape of the active site. Next is the efficiency of enzymes. Enzymes are very efficient. They are effective in small amounts. Just a small amount of enzyme is enough to bring about the conversion of substrates into products. But the substrate, the term substrate, is used to describe the substance upon which the enzyme acts. So enzymes act on substrate and convert them into products. So enzymes are efficient in small amounts. A small amount of enzymes will work on large amount of substrates. This is mainly because enzymes are not used up in the reactions that they catalyze, and so they can be used again and again. In fact, which brings us to the next property, that they're not used up in the reactions that they catalyze. So because of this, a small amount is sufficient and they can be used over and over again. Next is the reversibility of their reactions. Enzymes control reactions in either direction. Enzymes control reactions in either direction. So the same enzyme controls the reaction from left to right, this way, and the same enzyme will control the reverse process. Now, the direction of the reaction depends on external factors and the amount of substances on either side. So here in this example, this is the enzyme and that is the substrate. So if the reaction is from left to right, these substrates will be converted into two products, the two products. Now, if we have a high concentration of substrate, these substrates, then the reaction will proceed more from left to right, and the same enzyme will catalyze that process. Now, if we have a higher concentration of these substances here compared to this other one here, then the reaction will proceed from right to left, and the same enzyme will convert now these substances that become substrates into this one that will become the product. So if the reaction is moving from left to right, this is a substrate and this becomes a product. But if the reaction is proceeding from right to left, then these substances become the substrate and this one here becomes the product. So enzymes control reactions in either direction, and the direction of the reaction depends on external factors such as the relative levels or concentrations of the substances that it reacts or acts upon. So for example here, sucrase as an enzyme acts on sucrose to hydrolyze it into constituent monosaccharides, glucose and fructose. But if we have a high concentration of glucose and fructose in the system, then the same sucrase will catalyze the condensation of glucose and fructose to form sucrose. So it all depends on the prevailing conditions. It all depends on which substances are at higher concentration. So those are the properties of enzymes, and these properties determine how they function and gives them the ability to catalyze the reactions that they do control. Now, there are certain factors that affect enzyme controlled reactions, because depending on the prevailing conditions, an enzyme control reaction will proceed at a particular rate. These depend on certain prevailing factors, and these factors include: 1, temperature, the pH of the surroundings, substrate concentration, enzyme concentration, presence of enzyme cofactors and coenzyme, and also presence of enzyme inhibitors. We look at each one of them one by one, starting with temperature as a factor. Now you recall that enzymes are protein in nature, and being protein, the activities are influenced by temperature. Temperature in general affects so many factors in biochemical reactions. For one, temperature affects the rate at which particles do move, because the higher the temperature, the higher the kinetic energy of the particles and the higher the rate of reaction. So basically, enzymes react with substrates through collision. So the higher the temperature, the higher the rate of collision between the enzymes and the substrates to form products. Now, if we start at lower temperature, so the effect of temperature on enzyme controlled reaction can be illustrated in this graph below. At low temperature, this range here, the rate of reaction is relatively low. Why? Because at lower temperatures, the enzymes are said to be inactive. The enzymes are said to be inactive at low temperature. So at this point here, the enzymes are inactive. So between points A and B, the temperature is lower than optimum. But as we increase the temperature, the enzymes become more and more active and the rate of reaction increases. This will continue until the optimum temperature is attained. Now, for most enzymes, especially in the human body, the optimum temperature is between 35 to 40 degrees Celsius. This temperature produces the highest rate of reaction. If we continue raising the temperature and it goes above the optimum range, then a further increase leads to a drop in the rate of reaction. Why does the rate of reaction decrease beyond the optimum temperature? It's because beyond the optimum range, enzymes, being protein, get denatured. Enzymes get denatured. And when they get denatured, their effectiveness reduces. So as the temperature rises, more and more enzymes become denatured and the rate of reaction decreases and becomes less and less until eventually it may stop altogether. So at low temperatures, enzymes are inactive, so the rate of reaction is low. At the optimum range, there is the fastest rate of reaction because the enzymes are now activated. Beyond the optimum temperature, the rate of reaction decreases because the enzymes are becoming denatured. The next factor that influences the rate of enzyme control reaction is the pH. Every enzyme has an optimum pH. Every enzyme has an optimum pH because also being proteins, enzymes are affected by the surrounding pH. So different enzymes will have different optimum pH, which produces the highest rate of reaction. So if you look at these three enzymes here, for example, enzyme A has an optimum pH of about 2. This is what produces the highest rate of reaction. Enzyme B has an optimum pH of about 7, producing the highest rate of reaction, while enzyme C has an optimum pH of about 9.2 or 9.3 there, or about 9 thereof, that in which it acts fastest. So different enzymes have different optimum pH. So when the pH is either below the optimum or above the optimum, the rate of reaction decreases. For the same reason, like in temperature, that is the pH beyond the optimum, enzymes get denatured. So for enzyme B, pH below 7 is not conducive and the enzymes tend to get denatured. Above 7, the enzymes also progressively get denatured. So for efficient functioning, each enzyme requires a specific pH to work. Then next, look at the substrate concentration. Substrate, as we mentioned, is a substance that is acted upon by enzyme to be converted into product. Now, if we start at low substrate concentration and we increase the substrate concentration using a given amount or concentration of enzymes, then you're going to see that as we increase the concentration of the substrate, the rate of enzyme activity will also increase. But this will only go on up to a certain point. At a certain concentration of substrate, the rate of reaction will reach maximum, and even if we continue increasing beyond that maximum point, the rate remains constant. So this is the optimum concentration of substrate at a given concentration of enzyme. Remember, the concentration of enzyme is constant. So if we start with low concentration of substrate and progressively increase the concentration, the rate of reaction will increase. But up to a certain point, beyond this point, the optimum concentration for the substrate, even if we continue increasing the substrate further, the rate does not go beyond this particular point. And why is that? Now this happens because we have limited number of enzymes and therefore limited number of active sites. So beyond this point here, there'll be too many substrates for a constant number of active sites. So when the substrate concentration is increased, at some point all the active sites of the enzymes will be occupied. And this condition makes the number of enzyme molecules to be the limiting factor. This is what makes the number of enzyme molecules to be the limiting factor. So the only way to increase the rate of reactions beyond this point is not to increase the substrate concentration, but to make more active sites available by increasing the concentration of enzymes. So substrate concentration affects the rate of reaction up to a certain point. The more we increase the substrate, the higher the rate of reaction, but up to a certain point because of the limiting number of enzymes active sites. The next factor is enzyme concentration. Now, there are two ways in which you can look at the effect of enzyme concentration on the rate of reaction. One, by comparing the rate of reaction at different enzyme concentration. So if we continue increasing, let's say, the substrate concentration in two different setups, the rate of reaction will continue. But at a lower enzyme concentration, the optimum, the fastest rate will be attained at a lower point compared to enzymes at higher concentration. So if we pick, let's say, pick this point here, at this specific concentration of substrate, a lower concentration of enzyme attains a relatively lower highest rate of reaction. Whereas at the same substrate concentration, using higher concentration of enzyme, we are able to attain a relatively higher rate of reaction. So in this graph here, you can compare a difference in enzyme reaction. At a higher enzyme concentration, the highest rate of reaction attained is higher than at a lower enzyme concentration. Now, another way of comparing, looking at the effect of enzyme concentration on the rate of reaction is by comparing the rate of reaction, for example, time taken for the reaction to come into completion or to end, when you plot that against the enzyme concentration. So at different enzyme concentration, we measure and record the rate at which a reaction takes time, the rate or the time taken for a particular reaction to occur. And the best reactions are normally those that are marked by change in color. For example, you can use the time taken for starch to be broken down into sugars. When you combine starch and iodine solution, we get a dark blue color. So the presence of starch is marked by the dark blue color. So by adding different concentrations of enzyme, we can monitor how long it will take for this dark blue color to disappear, because when all the starch is broken down into starch, then sugars do not react with iodine solution, so this dark blue color is going to disappear. So we take different concentrations of enzyme and mix them with equal volume of starch solution, and we monitor how long it takes for the dark blue color to disappear. Then we plot the findings in a graph like this, and we see that when you plot the graph, the curve is going to slope downwards like this, meaning that the higher the concentration of enzyme, the faster the rate of reaction, indicated by the shorter the time it takes for the dark blue color to disappear. The higher the concentration, say at this point here, a relatively shorter time will be taken for the dark blue color to disappear compared to a relatively lower concentration at this point here, when the time taken is going to be more. So this way also we can relate and look at the effect of enzyme concentration on the rate of reaction. From this graph you can see that the higher the enzyme concentration, the faster the rate of reaction, indicated by the time taken for the reaction to come into completion, because the time taken is inversely proportional to the rate, and the faster the rate, the shorter the time to be taken for the reaction to come into completion. Then you also have other factors that do affect enzyme controlled reactions, and these are enzyme cofactors and coenzymes. Now, enzyme cofactors are non-proteinaceous substances which activate enzymes. There are many enzymes that cannot function in the absence of certain other substances. These other substances are known as cofactors. So the absence of these cofactors will mean that the reactions cannot take place. And again, the lower the concentration of the cofactors, the lower the enzyme activity, and hence the slower the rate of these enzyme controlled reactions. Now, most enzymes, however, will not work without these cofactors. Some of these cofactors are metallic ions, but some of them are metallic ions like ions, magnesium function as cofactors, zinc ions also function as cofactors, copper, and many others. While you also have other cofactors that are organic, though non-protein in nature. These organic compounds that are non-potent in nature that act as cofactors are known as coenzymes. Coenzymes are mainly vitamins. Vitamins are non-protein molecules which are organic in nature, which are necessary for the activities of certain enzymes. So, coenzymes are usually derived from vitamins. So, alongside the ions of many metallic substances, vitamins make up cofactors, and many enzymes will not function in the absence of these cofactors. Then the last factor that affects enzyme activities are the enzyme inhibitors. Enzyme inhibitors are substances which have similar molecular structure and resemble the substrates. Hence, they can also bind to the active site of the enzyme, and in this way, they slow down or even stop enzyme activity. There are two types of enzyme inhibitors. One, competitive inhibitors. These competitive inhibitors attach themselves, this is an enzyme, so they attach themselves to the active site, though temporarily, of enzyme. They attach themselves to the active site of the enzyme, though temporarily, but they normally stay longer, and though they are not affected by the enzymes, but they attach themselves onto the active site, and by taking up this active site, they prevent, say, the actual substrate. Let's say this is the actual substrate that should be on the active site of the enzyme. The fact that this inhibitor takes up the active site, it prevents this substrate from combining with the enzyme, and therefore the rate of the reaction is going to be slowed down. Now, this type of inhibition can be overcome by increasing the substrate concentration. If we increase the concentration of this substrate, then that means that there will be higher chances of this substrate getting to the active site before the inhibitor does. So one way of overcoming the competitive inhibition is by increasing the substrate concentration. So if we increase the substrate concentration, we increase the chances of the substrate getting to the active site before the competitive inhibitor does. So in competitive inhibition, the inhibitor and the substrate compete for the active site of the enzyme. Even though the inhibitor is not affected by the enzyme, by taking up the active site, it prevents the substrate from getting there, and therefore it slows down the conversion of the substrates into products. The other type of inhibition is known as non-competitive. Now, in non-competitive inhibition, the inhibitor substance attaches itself permanently onto the active site, and in this way completely blocks the active site and prevents the enzyme from interacting with the substrate. As more and more active sites are blocked permanently by the inhibitor, the rate of reaction slows down and eventually the reaction may stop. You see, in non-competitive inhibition, the inhibitor does not really compete with the substrate because it stays in the active site permanently. It actually destroys the enzyme. But in competitive inhibition, the inhibitor interacts with the enzyme, though temporarily. So it comes to the active site and goes, just like the substrate. But in non-competitive inhibition, the inhibitor attaches itself permanently and in the process destroys the enzyme. In fact, most non-competitive inhibitors are very potent poisons that affect living organisms, like cyanide. Cyanide functions as a very strong non-competitive inhibitor that affects respiratory enzymes. This is how cyanide is able to stop the release of energy through respiration, and without energy, cells cannot function they die, and eventually the entire organism dies as a result.